Production of recombinant proteins. We should remember first what's a recombinant protein. Okay, normally encoded in a plasmid, but can also be inserted into the host genome from which it's going to be highly expressed. And the other important part is the hosts. We can see that we have prokaryotic, mainly bacteria, but also we have eukaryotic. Among the eukaryotic, the most common ones are mammalian cells, but they drive certain disadvantages such as the price and the complicated systems of expression, besides to a low tolerance to changes, low cell tolerance. And recently, yeasts have been an incredible source of for heterologous expression. We can see that different organisms used as hosts require different kinds of protocols in order to promote that high expression. In the market, there are more than 200 protein drugs products. The production it's crucial for both the development of new protein drugs, but also for the structural determination of the targets. All times, recombinant proteins were produced in low concentrations, and there were big problems because most of them were intracellular, so they required tough and complicated downstream processing, where most of the highly expressed protein couldn't be solubilized. But the arousal of recombinant DNA technology enabled a very sophisticated genetic repertoire of techniques to modify the host, modify the plasmid carrying the recombinant the protein, and have been able to or to place where the protein is going to be expressed and delivered, either in the periplasm, outer from the cell, inside the cell, etc. As in not only the past but nowadays, most of the recombinant proteins are produced in prokaryotes. They require that gene expression is gonna be achieved while cells are growing. So we have to look for high levels of gene expression and high cell density cultivation. And you can take a look here about which has been the main products produced as recombinant proteins. The process for its production, it's as you can imagine an upstream where you're generating those vectors, those expression vectors. You have to take a look for promoters. You have to reach strong indocible promoters that are tightly regulated to avoid leak expression. You have to ensure an effective replication of the plasmids, and you will have to take care of if you include any kind of tag which will ease the solubilization, translocation, and purification of the proteins. Afterwards, this upstream task, we go to the fermenter, normally produced in batch and in stir tanks, but depending on the kind of protein, where it's gonna be released, inside or outside the cell, and the kind of host you're employing, this might change. And for sure, downstream processing, it's one of the most important parts of the whole process. Here you will have to reach high levels of purity of the protein, and these proteins normally should be functional later. Must be functional later. So you should be sure that the techniques that you're going to apply for the downstream processes do not compromise that functional capacity. As we said, most of the recombinant proteins available in the market are from a bacterial using bacterial hosts, but mainly E. coli. Why this one's because they grow fast. We have a wide knowledge and protocols of for them. They can express heterologous proteins at high levels, even at 50% of the total cell protein content, but they also present several disadvantages. As prokaryotics, they have no Golgi apparatus and reticulum endoplasmatic reticulum, so there are no post-translational modifications. And you know that many eukaryotic proteins require glycosylations, phosphorylations, etc., which cannot be achieved in E. coli. So you can have unfunctional proteins due to that. Besides, there are certain eukaryotic domains that cannot be correctly expressed in E. coli because it cannot read the E. coli ribosomes cannot read properly these certain domains. As a halfway between the very expensive and complicated and sensitive mammalian cells and the highly different bacteria, the arisal of yeasts being employed as hosts for recombinant proteins has been a major breakout. Why? Because these are eukaryotes, so that you can have these post-translational modifications, but they're very simple, and there are many genetic tools to modify them, to manipulate them, and to finally achieve a high cell expression to accompanied by a high cell density. Sometimes mammalian and insect cell cultures are required for specific protein products, and this mammalian cell or this requirement of using this kind of host normally imply that the downstream processing is gonna be normally easier or more, not necessarily easier, but more directed to specific properties of the tag protein. One of the main recombinant proteins or one of the main products of interest in the pharma industry, and especially these days where all scientists are looking desperately for treatments for coronavirus and so on, is the production of vaccines. Making human vaccines available on a global scale requires the use of complex production methods, very complex methods, and meticulous quality control and reliable distribution channels to ensure that the products are potent and effective at their point of use. So it's not only complicated the synthesis of these vaccines, but also to reach the legislations, the high quality control, and ensure that the distribution of these vaccines are going to respect the efficiency or the effectivity of the synthesized vaccines, and also are going to be effective and as potent as they should be upon the population. This might be one of the main reasons why there's a very high demand, a very importance on the production of vaccines, but there's a very low number of actually producers that have thrived on the vaccines production. In vaccines, we normally have bacterial vaccines, but also viral vaccines. Okay, vaccines against certain protozoa have been in development. For example, against Plasmodium, malaria, and in the production of these vaccines normally involved an initial phase of formulation and stabilization, and then high production for the conservation of this vaccine, or to increase its potency. It's normally mixed with adjuvants, and then There's also a final step of a delivery device design of how it's going to be administered, and in order to ensure and to keep and maintain its potent and effective form of the product. You have I don't get into the types of bacterial vaccines because you already know from microbiology, but I will want to highlight that for different viruses, for example, you can use kinds of different types of cell types and different forms of different kinds of vaccines will be produced. The pipeline of the production of vaccines, you can check it here. Not only bacterial ones but also viral ones are submitted to a very highly controlled production with multiple steps, and as you can see here, the downstream processing is super intense. You not only have to reach high levels in the fermenter, normally in small fermenters, to avoid contaminants and optimize the yields of the antigen and so on, and a tight monitoring and thorough control of items such as oxygen dissolve, pressure, etc. I was mentioning that downstream processing is very intense and normally requires the highest levels of purity. So the most sophisticated techniques are the ones that are going to be implemented. If you can take another look of, for example, this is what Merck does for the production of of a viral vaccine, okay, and different bioreactors and these different steps of downstream with chromatography. And with this, we finish the unit. If you want further information about the vaccines, I have uploaded, because I think it's kind of high and prior interest in current days. I have uploaded two papers that are actually a review about the difficulties of producing vaccines that go from several months, if we know the microbe and the environmental conditions where we're going to find the microbe, to nearly 3 years. Many factors such as the target population, the mechanism of action, the environmental conditions of the microbe, the manufacturing capacity of the companies, many factors are gonna influence the optimal production of these vaccines.